We have seen the normal reproduction, that is the sexual reproduction in angiospermic plants. We have also seen the alteration of generation, that is from gametophytic haploid stage to sporophytic diploid stage and vice versa in an angiospermic plant. This is a clear alteration of generation. But in some cases, there are examples where seed formation takes place without fertilization, or there is the alteration of generation is deviated from the normal cycle. This deviation is referred to as apomixis. So how do we define apomixis? Apomixis can be defined as a substitution to sexual reproduction, where the seed formation takes place but without undergoing meiosis or fertilization. There is no reduction division, and hence engamy or fertilization does not take place. Apomixis is actually asexual reproduction which mimics sexual reproduction. That is, the formation of seeds take place, which is the end product of sexual reproduction. Here seed formation is taking place, but fertilization or reduction division is not taking place. Apomixis can be of two types: vegetative reproduction and agamospermy. Vegetative reproduction we have studied in the previous chapter, and we have also studied in smaller classes the different modes of vegetative reproduction, like the roots are there, shoot system is there, then there are runners, then there are offsets. These are known as vegetative propagules. Actually, we call them as vegetative propagules. Which help in propagation, vegetative propagation. So this is a clear asexual mode of reproduction or asexual mode of propagation. We did not go in detail. Now let's come to agamospermy. Agamospermy is formation of the gamete or the development of the gamete without undergoing fertilization. The formation of seeds without undergoing fertilization, that is, seeds made by apomixis, are known as apomictic seeds. So let's come to agamospermy. Agamospermy can further be divided into parthenogenesis, and adventive embryony. Now all of us know what is parthenogenesis. The formation or the development of the female gamete without fertilization is known as parthenogenesis. Parthenogenesis, all of us know, can be natural also and can be induced. Now parthenogenesis are of two types: haploid and diploid. Haploid parthenogenesis, if the embryo sac develops without fertilization, that is, the eggs from the embryo sac, they develop without fertilization, then it is known as haploid parthenogenesis. The seeds formed in haploid parthenogenesis are sterile. Now in diploid parthenogenesis, any diploid tissue other than the embryo sac develops into embryo is known as diploid parthenogenesis. Now these diploid tissues can be the integuments or the nucellus. Now parthenogenesis can again be developed or divided into diplospory, and apospory. In diplospory, the diploid cells, the diploid tissues, other than the embryo sac or the egg cell, helps in the formation of the embryo. Now these diploid tissues can be the somatic cells like the nucellus, integuments. They develop into egg cells which gives rise to a diploid embryo. In diplospory, the megaspore mother cell does not undergo meiosis and forms the megaspore. The megaspore forms the embryo sac which is diploid in nature. The egg cell which is present in the diploid embryo sac multiplies and forms the zygote which gives rise to a diploid embryo. In apospory, it is the development of the diploid embryo sac from the normal plant cells which are diploid. Now these cells can be the nucellus or the integuments which gives rise to a diploid embryo sac. The diploid egg cell which is present in the embryo sac which is diploid in nature gives rise to zygote without undergoing meiosis. Now this zygote gives rise to a diploid embryo. Now both in apospory and diplospory, diploid eggs are formed. Diploid embryo is formed which is actually an unusual type of reproduction. Now let us come to the adventive embryony. In adventive embryony, the embryo develops inside the ovule from any diploid tissue other than the embryo sac. These diploid tissues are sporophytic in nature. This means a sporophytic generation giving rise to a new sporophytic generation. The sporophytic tissues, they form a bud and start behaving like an embryo. So this is also known as sporophytic budding. Important is that in adventive embryony, the embryo sac is not formed. Now let us come to polyembryony. This is also the formation of more than one embryo inside the ovule. Best examples can be taken from the citrus fruits. The seeds of citrus fruits clearly show polyembryony. So we come to polyembryony. This is also an unusual feature which is found in the angiospermic plants. Polyembryony means the involvement of more than one embryo or formation of more than one embryo inside the ovule is known as polyembryony. Here one should make clear that fertilization and gamete formation takes place in polyembryony, but only thing is that more than one embryo is formed inside the ovule. Now there are many reasons why polyembryony takes place. It can be the presence of more than one embryo sac, that is more than one embryo sac. More than one embryo sac. Then second may be the synergids and the antipodals. They also start behaving like the egg cells. So one reason of polyembryony was that more than one egg cell was present in the embryo sac. So there can be more than one egg cell in the embryo sac. The second reason can be the synergies and the antipodals. The synergies are present besides the egg cell, and the antipodals, they are present just opposite to the synergies in the chalazal end. Antipodals, they start behaving as egg cells. Another reason can be that the zygote can be broken into fragments. And each fragment develops into embryo. And each fragment, all the fragments, they will start developing into embryo. Develop into embryo. Now we have seen that the zygote can be divided into fragments, and each fragment, they start behaving like a embryo, develop like a embryo. Another case can be that more than one embryo sac is present. More than one embryo sac Now we have seen Apomixes and Polyembryony. Both Apomixes and Polyembryony are very helpful in modern agriculture. They give rise to hybrid seeds which does not lose their character.